So hello, Francesco Pignatti. Very happy to have you here. So to present yourself quickly, you're a medical doctor. You have also training in biostatistics, and you're working at the EMA, so the European Medicine Agency, in the Oncology, Hematology, and Diagnostics department, mostly on the approval of medical products. So we are very happy to have you, and we have a few questions on your activities. So first, could you present your activities at the EMA, please?
Yes, hi, and thank you very much for the opportunity to be here. I've been at the agency for almost 25 years now, and I've pretty much always worked in the office of oncology, which is an office which supports our scientific committee. Our scientific committee, I mean mainly our Committee for Human Medicinal Products. This is a committee that evaluates new drug applications, for example, and then decides whether they should be approved or not, and what should be the label, what should be the therapeutic indication, what should be additional studies to be conducted post-approval, and so on. And at the agency, we have a small team of scientists who support this committee in their decisions. Aside from these activities, I'm very interested in all aspects of cancer research and methodology, and also in benefit risk assessment, which is a central part of our decisions.
First, in personalized medicine, there is a big deal about approving compounds, so targeted therapy mostly, or targeted immunotherapies, or other type of therapies, either in a specific tumor type, or there are some example of tumor agnostic approvals. So what is your experience on this different way to approve the compounds, such compounds?
Well, thanks a lot for this question, because we talk about this type of drugs a lot. You can imagine, on one hand, personalized medicine gives a lot of hope, a lot of uh, interesting drugs that are being developed, but at the same time, a lot of challenges, because as we target more specific group of patients, so the numbers of patients dwindle, and then it becomes more difficult to do, for instance, randomized controlled trials. And then there is a big discussion of what type of uncertainties you can accept. And I think you've rightly divided them into two groups. Perhaps the ones we are most used to seeing are when all of a sudden, let's say, a large indications like non-small cell lung cancer becomes fragmented because there is a recognition of driver mutations that can be targeted by specific drugs. And this often poses tremendous advantages from the point of view of how much we understand about the drug, how it works, and that certainly helps in the decision. But also, as I mentioned before, smaller trials where one can collect the evidence that we need for approval. And in those situations, we are often helped because there is a very high activity. And so randomized controlled trials can be run even in smaller populations relatively efficiently because there is a large effect. And to observe a large effect, you just need small sample sizes. Where it gets a bit trickier is if the activity is such that randomized controlled trials are, for instance, impossible to conduct because of loss of equipoise. You have sometimes these very dramatic responses, and then patients, clinicians will not want to randomize patients. And in those situations, often companies submit applications just based on single arm trials, or let's say non-randomized controlled trials. And that poses challenges from an evidentiary standard point of view. Luckily, in our regulation, we have what we call the conditional marketing authorization, where if there are important uncertainties due to non-comprehensive data, for example, because the randomized control trial is missing, you are able to conduct studies post-approval and to impose companies to submit such studies post-approval. And then you have this conditional marketing authorization, which are going to reassess the benefit risk on a yearly basis based on these new data. And this has been regularly used for this type of approval, not without controversy, because obviously approving drug when the data are not controversial, as not comprehensive, is controversial because you're allowing some more uncertainty. And I guess the important thing there is that we are even more transparent and even more clear about what these doubts are and how we made our decision. Okay. Now, I'm just going to briefly touch on the other type of approval, which of drug, which you mentioned, the so-called agnostic drugs. I prefer to talk about biomarker-driven indications or something like that, which reflects the mechanism of action that we know much. In reality, these are drugs for which we know much more about, and where we know that what matters is not, let's say, the traditional anatomical taxonomy for selecting patients most likely to respond, but something else, something like a driver mutation that would act similarly across anatomical or other type of factors. And there again, we run into problems of sample size often and inability to run randomized controlled trials, and kind of the problems become the same as the other type. But there is this added element of trying to detect an interaction for the different tumor types. Again, it's a delicate balance, and I think what is key, what we strive for, is to, if we have sign of very dramatic activities, to make sure these drugs are approved quickly for the benefit of patients, but at the same time that we're crystal clear about what are the standards of evidence that underlie these decisions.
In the first situation you described, when there is a dramatic effect of a single compound in a specific cohort of specific tumor type, are there examples of approval of compounds with small numbers of patients? And in this case, if there is conditional approval, how long do you agree, or do you advise to wait for post-conditional approval validation? What are the scenario in this case?
You know, to be honest, there is no single scenario that is going to be really a prototype. When we assess this application, we always looked at the totality of the evidence. And so maybe it's not the number of patients in the clinical trial, but it's the very strong pharmacological rationale that is going to drive the thing, or it's going to be experience for the same drug in other indication, which is going to provide, I don't know, huge reassurance on the safety profile and so on. So I cannot pinpoint to you a secret formula that, I don't know, you need 100 patients and in your efficacy set and in your safety database. So, and I think this is not appreciated enough. People think that this is about reaching a magic threshold of, say, 40% response rate. Sutent, sunitinib in renal cancer was our first conditional marketing authorization in 2006, and it had roughly a response rate of this kind. So that stuck to people, like, if we have this percent, then a conditional marketing authorization is possible. No. That is just one piece of a much bigger puzzle, and it's kind of the wrong way of looking at it if people go away thinking that, you know, as long as you have a few hit a few parameters, that means an approval. No, it's a complex decision that takes into account all of the totality of data.
Yeah, I see. Okay. And for example, it drives me to drop trials, which is our main activity in PCM for you and Primrose. So drop trials are repurposing compounds that have been already approved or already assessed in previous trials. So for which we already have quite a large amount of safety and efficacy data. The drug trial proposed to repurpose those compounds in new population of patients with specific cancer or even new biomarkers. In this situation, there have been a success story recently in the Netherlands where there were a cohort of patients with MSI high tumors finally derive in an approval in the Netherlands and reimbursement for MSI high tumors. So my question is, could do EMA have similar process for repurposing? And as EMA and your team and in your opinion, could this model of repurposing with mono arm trials could be a good design for new approval for a compound?
Well, I think this is a great question, and I have to warn you that I'm answering here with my personal views because we don't
Okay.
have an agency position on this design of trial or this type of strategies. What I find particularly interesting is not just the attempt to match a certain patient with a certain tumor type or a certain mutation with a particular drug, which is really harvesting all the benefits of personalized medicine, but also the fact that we are doing this within a controlled context of evidence generation so that this is an experience that can be used to generate useful data for also for other decisions and other decision makers. So I find this extremely interesting. And in general, although off-label use of approved drugs is not something we regulate, it's something that is for the countries, and then they need to think, how they give these treatments and how they are reimbursed, etc. It's not something we get involved in at all, but we do recognize that in a number of situations, there are approved drugs that have a use that could be rational and could be even demonstrated at times. And people feel that there is a need for having a formal uh, way of repurposing these products. And so together with the European institutions, we are looking at ways to facilitate this repurposing. And there has been a pilot launched at the level of our scientific advice, where we provide, let's say, an assessment of the strategy for generating evidence under this repurposing framework that could then lead to a marketing authorization. So again, for the drop trial itself, I cannot comment on whether this strategy will be successful or not, but that aside from finding it personally very interesting, there will be mechanisms by which these type of efforts can be qualified from a regulatory perspective, and then to ensure as much as possible that they allow formal regulatory scrutiny and potential marketing authorization applications to take place. So it's very much in our focus and a very interesting initiative.
In practice, is it better before to launch such repurposing trial to come to your team or the EMA to be sure we are in line with these mechanisms of regulatory steps for up to approval, or is it a post post trial analysis that you perform? What is the best way to go in practice?
Yes, I think I would always recommend this level of interaction between regulators and academia or whoever is conducting this type of trials, because apart from the benefits, there are also the challenges, and we discussed some of them. Lack of randomization makes this data difficult to interpret. also, if these are run and as platforms of multiple agents, there is a temptation of comparing um, different arms of such trials, and then that adds a complexity adds many complexities from the point of view of statistical aspect. But it doesn't mean it doesn't have to be discussed. It cannot be discussed. On the contrary, we are there, and the format can change. So we have a number of different ways you can interact with the agency, some more formal, like what we call a qualification, where a whole system or a whole set of evidence is qualified for a certain purpose, or less formal, more discussion meetings or collaborative efforts where we could, participate jointly in a European project trying to develop methodology that will then benefit all researchers. So what I'm saying is that there is an array of different ways to interact, and you definitely can contact EMA, myself, other EMA colleagues to really understand what are the many opportunities that we offer to maximize these interactions.
Okay, thank you very much, Francesco Pignatti. I think we covered a lot of topics of great interest, and I hope this would also be of interest for our audience. So thanks a lot for your answers.
It's my pleasure. Thank you.